Good afternoon. I'm Eric Isaacs, president of the Carnegie Institution for Science, and welcome to our Capital Science Evening program today. Really honored to once again, as we do every year, partner with the Kavli Foundation, their new president, Cynthia Friend, the Norwegian Embassy of the United States, and the Norwegian Academy of Science, and letters to feature a Kavli Prize laureate in this series. Today's program, we're all looking forward to greatly, is a conversation between Professor John O'Keefe, who received both the Kavli Prize and in nanoscience and the Nobel Prize in the same year in physiology for medicine in 2014, and also the Emmy-winning journalist and a friend of ours, Frank Sesno. Dr. O'Keefe is the professor of cognitive neuroscience at the University College London in London, and was the inaugural director of the Sainsbury Wellcome Center for Neural Circuits and Behavior. And his research focuses on a very interesting topic that I think we all have spent some time thinking about in our own daily lives, which is on explaining the neurological underpinnings, how the brain works on one thing, which is how to orient ourselves. This is a question, as I said, you probably have thought about yourself, and how does your brain figure out where you are in space? And very interesting problem. Embedded deep in the brain's temporal lobe, it's driven by the hippocampus. It plays a major role in learning, and Professor O'Keefe's research has been critical to revealing its importance for this kind of spatial memory and navigation. And the loss, of course, is prominent in diseases, including Alzheimer's and many others. Several decades ago, he made a fundamental breakthrough, discovering a particular cell type in the hippocampus, which helps us form the mental map of space. So this discovery of so-called place cells, which enable the hippocampus to serve as a cognitive map through which we understand our environment and where we are, has been extremely important both then and now. This whole field of cognitive neuroscience benefited greatly, in fact, may have really developed a great deal from this important milestone, this important discovery. And since that time, he has continued to reveal the signaling processes between these cells to really try to untangle the entire spatial memory apparatus that exists down in the hippocampus. Professor O'Keefe earned his doctorate in physiology, psychology from McGill University in Canada, and following a bachelor's degree from City College of New York. He then joined the University College London as a US National Institutes of Mental Health postdoc fellow, and never left. He stayed there since. He's a fellow of the Royal Society of the Academy of Medical Sciences and a member of the US National Academy of Sciences. And in addition to the Kavli Prize and the Nobel Prize, he is a recipient of the Feldman Foundation Prize, the Grauermeyer Award, the British Neuroscience Association Award, the Federation of European Neuroscience Societies European Neuroscience Journal Award, and the Louisa Gross Horowitz Prize. His work is so foundational that it cuts across many boundaries between science and philosophy. And it is really an honor to have him with us today and to hear what he's going to say in the next hour. The event today will be structured as a conversation between Professor O'Keefe and Frank Sesno. Frank is the Director of Strategic Initiatives at George Washington University School of Media and the television project that highlights innovations in sustainability. Prior to this, Frank was the school's director for 11 years and spent 20 years at CNN, where he served as the White House correspondent, anchor, and Washington bureau chief. So I know everybody is looking forward to hearing this fascinating discussion between Professor O'Keefe and Frank Sesno. So please join me in welcoming the two of them here to our evening.
Eric, thank you very much. It's delightful to be here, and it's delightful to be with you. And thanks to Carnegie, thanks to Kavli, and thanks to John O'Keefe. And John, welcome to you.
Thank you very much, Frank. It's great to be here, and I'm very honored to be asked to give this lecture.
It's going to be great. And a couple of things right off the top, John. We have more than 1,000 people register for this event, so you have a huge audience, even though you can't see them, unfortunately. And we welcome everybody. And the other thing I want to say is we will have some polling, some questions that we'll put to the audience for those of you who've been in these discussions with us in the past. You know that occasionally we'll ask you a few questions or a question. You can register your response, and then we'll get our guest to respond to that, and we'll go from there. John, where are you coming to us from? Is this your office, your home? Where are you right now?
Well, this is a corner of my living room, which I've been using as an office for the last couple of years during lockdown. I like it a lot. I feel very comfortable here, and it's good to work here, and it's also good to talk to you from here.
And you're in London in the UK now, right?
Yes, I am. Have been
Your
as
health has been good. Everything's okay.
everything's good. I've had 2 jabs of the Pfizer, and looking forward to a booster shot. Yeah.
Well, I'm really looking forward to the conversation. As I say, what we want to do here is explore you and your science, engage our audience a bit. John, what you've done with your research in hippocampus, what you tell us about ourselves is fascinating. We'll talk a lot about that, but how you got here is a story in and of itself. I mean, you were building airplanes on Long Island and studying filmmaking before you got into this. So how does this incredible trajectory lead you to where you are today?
Well, yes, I'll tell you a little bit of what I term somewhat jocularly my misspent youth, and how I went by a very circuitous route to neuroscience. I grew up in the South Bronx. Went to the local parochial school, and at the end of 8 years there, I was lucky, or perhaps in retrospect, unlucky enough to win a scholarship to a very prestigious Jesuit school in Manhattan. I say unlucky because the major studies there were classics, Latin and Greek. And for reasons which still aren't clear to me, although it was clear, I think, to the Jesuits who taught me, I never really could get a handle on Latin and Greek. And so after several years where I failed to learn, for example, Latin verbs and so on, I earned the sobriquet of stupidissime, which I don't think you need very much Latin to translate. And was told I wasn't actually I wasn't very good college material. And so I was cast out and had to find a job. And I took several jobs, clerical jobs. I worked feeding Hollerith cards to an IBM machine. We already had these monstrous IBM machines. And I learned there that if I were ever going to have a computer of my own, it would have to be smaller than me, because these things were the size of a room. But eventually, and at the same time, I was taking engineering. I decided to take something which was about as far away from classics as I could get. So I took aeronautical engineering at NYU. Through that, and through my contacts there, I eventually got a job working for Grumman Aircraft. As you say, I went on Long Island.
Yeah.
So I was commuting from the Bronx to Long Island, and then back to NYU and back to the Bronx. It was quite a strain. While I was at Grumman, I slowly decided that I didn't really want to make airplanes, which were primarily warplanes, for the rest of my life.
Because this was during this was in the middle of the Cold War too, right?
This is in the middle of it. This is 1957 to 1960. And I also started taking courses even at NYU. I started taking courses in philosophy and the history of science and things like that, and began to think about the idea that a lot of the questions that were coming up in these courses seemed to me ones which could actually be handled if we could get access to the brain. So I already started to think about how looking at the brain might help us to solve some of the classical problems in philosophy.
Such as?
Well, such as consciousness or perception or identity or cognition in general. So as I thought about it, I finally decided, I took my courage in my hands. I sold my Ford Fairlane convertible, and I was lucky enough to get into City College of New York, which at the time was the only free college, tuition-free college in the United States. And I was like a kid in a sweet shop, because they had a policy where you could take more or less any course that you wanted. And I did. I took courses in just about everything. I continued my courses in physics and engineering, but I took courses in philosophy. I took courses in psychology. As you pointed out, I took courses in film studies, advanced English, and so on and so forth. And I was having a ball. And I thought, this is the life that I really wanted. I was poor, but I was actually, felt I was a scholar manqué. Until about 3 years later, I got this call from somebody called a dean. I had no idea who a dean was. And he said to me, you know, it's people like you who are going to ruin this place. Because I had taken literally hundreds and hundreds of credits. And he said, you have enough credits to have any one of several degrees. Take one and get out. And I thought, oh, this is the end of my whole career. While I was at City, I had taken lots of courses in psychology, as I say. There was nothing called neuroscience in those days, but I had taken courses in psychology, and I had the good luck of finding a laboratory where we were doing pigeon work, and I was addicted to it. I knew that this is what I wanted to do.
Where you were doing pigeon work.
Yeah, we were working on pigeons. A fellow called Phil Ziegler, who had just come from Wisconsin, had set up a lab looking at pigeon behavior.
And you were looking at their exploratory behavior.
Their exploratory behavior, exactly. So I was very, I also, when I was at City College, I learned about Donald Hebb and his book, The Organization of Behavior, which had actually set out as a thesis exactly what I was interested in. Donald Hebb actually argue that you could understand things like perception by looking at brain cells and brain circuitry. So I was very much taken with that. And so I went to one of my psychology tutors and I said, I'm gutted. I don't want to go back to the world out there, the cruel world. What do I do? So he said, that's easy. There's something called graduate school. So I then went and applied to several graduate schools, but particularly to McGill University, where Donald Hebb was still working. And I was, again, lucky enough to get into McGill, and I went there. And I was studied for 4 years, and I was fortunate enough to have a mentor, Ronald Melzack, who had gotten a large grant by those standards from ARPA to study new ways of looking at the brain. And so it was there that I developed many of the techniques for putting electrodes into the brain of animals and studying the cell firings and to seeing what they correlated with. At the time, I have to say, many people thought this was just not the way to do it. They said the brain is so complicated, looking at single cells and just looking at what the animal is doing is never going to tell us anything. But I there studied cells in a part of the brain called the amygdala, and I made some sense of them.
I was noticing, by the way, John, if I may, that your work with the amygdala brought together your work with engineering, because you were working with microwire and electrodes,
yes,
and preamplifiers, and all
yes,
this
very
stuff.
good point. So I had the advantage of going into the field at the earliest stages, and I could bring to bear many of the skills that I had learned in electrical engineering to design small amplifiers to enable me to look at these very tiny signals as animals were moving around and actually behaving fairly naturally.
So let's talk about that. And that brings us to a point where we can ask our first question, our first poll of the audience, because your work went from the amygdala to the hippocampus. So let's see what people know about the hippocampus. So if we can put the question up for everybody, there you go. The hippocampus in the brain is principally responsible for, and you've got either certain physical endurance, certain types of memory, aggression and violence, or our sense of humor. So go ahead and put in your response to that. And we're getting a lot of responses very quickly. And while we get these in, John, very, very quickly, what you started with pigeons. Is there anything we learned from pigeon brains about the human brain? Or are these brains at all alike?
In fact, they're very different in many ways. The structure is very different, but we know that there are many behaviors which many animals have, most vertebrates have, and in fact, spatial navigation and spatial learning is one of them. So we didn't study that at City College, but I was already aware of the various commonalities between avian brains and mammalian brains.
Really. All right. So I think we've got a pretty good sampling of the audience from this question. So if we can put the response up so everybody can see that now. Here's what we got. We've got 87% say certain types of learning and memory, John. So that's pretty good. And Eric's probably pretty happy about that because I think he gave a little hint in his opening remarks. Some people say 10% aggression and violence. Might even ask you about that if we get any of that from there. 2% physical endurance, and 1% have a sense of humor and say it was our sense of humor. So what do you make of that? 87% have some knowledge.
That's terrific. That's terrific. And you may have kind of blindsided some people because the amygdala, of course, is one of the areas in the brain which is associated with aggression and violence. So they might have thought you were asking about the amygdala. I think it's terrific. And I'm not sure whether that, for me, that's good news that I've had some influence on the general audience, or whether it's bad news that I probably will not have a job in the next couple of days.
I really don't think you have to worry about that.
I actually just only started studying the hippocampus when I went to University College London. I took most of the technology there and decided that the hippocampus, which we knew had something to do with learning and memory from the work of Brendan Milner at McGill, was probably a good place to look. And I was looking, using the same technology, looking at cells and animals moving around for memory cells, essentially. And I looked for many, many months with one of my students, Jonathan Dostrovsky, and we didn't see any memory cells, or at least not obvious memory cells. But what we did see, and it took a while to appreciate this, were cells which seemed to have something to do with where the animal was in the environment. And that was the point at which I had this enormous aha, elateness moment in which I
referred to it as an electrifying day.
Electrifying moment. And it was a moment to begin with, but it actually lasted all day because I realized that if these cells were actually something to do with space and a way in which the brain was coding for space, that would be an enormous finding. You never know when you make these discoveries whether they're going to hold up, and you always have to have in the back of your mind that they have to be verified and replicated and so on and so forth. But a discovery like that actually I thought was going to be very, very important. Part of the reason for that was there was a man in California called Edward Tolman who had actually postulated that the brain of mammals was actually designed to create what he called cognitive maps, ways in which little rats could find their way around an environment, and using more than one strategy to find their way. It was a flexible way of finding their way around, not just going to the corner and turning left, but going by any available route. I also, of course, remembered my philosophy and remembered my studies of Kant. And Immanuel Kant, the 18th century German philosopher, had said, there are some very, very basic functions that the mind carries out. And they're so basic that they probably are there before you have any experience. And I guess we would interpret that now you're born with them. And he said space was one of them, time was another one, causality was another one. And so I began to think, if this was what we were looking at, some sort of a mapping structure, it would be really important and seminal to a lot of our understanding of the hippocampus itself and the rest of the brain.
And I know you have some slides to walk us through a little bit. So why don't you show us this so we can get a sense of what is happening or what you were seeing in the hippocampus as you researched and as we try to find our way around.
Okay, so I've got two slides to take you through on this in this section. So what I'm going to show you is what we actually saw using modern technology and modern ways of analyzing the cells. I have to say, in those days, we were, although we had some electronics, we were really reduced to looking at the animal and listening to the cells to trying to make out some correlation between what the cells are doing. So we're looking at the hippocampus, as you can see on the upper left-hand side there, and that shows where it is in red in the human. And as I say, we knew it had something to do with learning and memory because Brendan Milner had studied a famous patient who, when he lost his hippocampus through a surgical operation, actually was amnesic. He couldn't remember what had happened to him in the past, and he couldn't really learn new things as well. If you look in the middle slide on the top, that's what the hippocampus in blue looks like in the rat. And what we did is we put electrodes into that under deep anesthesia and allowed the animal to recover, and then saw what the cells were doing. Now, I had realized that at McGill that there were many cells in the brain which are silent most of the time, and they only became active and started to wake up when they had a job to do. And that's why when I looked in the hippocampus and saw that there were lots of these cells, I became very excited. If you see on the upper right, it gives you a simple idea of how we do these experiments. There's a mouse or a rat in a little box, and we're taking brain signals from its brain, and we're looking at where it is with the camera, and so we know where it is when the cell fires. And if you look at the lower left here, this is a cartoon of what we typically see when we do this in the hippocampus. You can see an animal running around, and this is real data, but I've made a cartoon of it. And every once in a while, one of these cells fires, and you can see the little red dots are left where the cell fires. And you can't hear the pips, but if you were listening to an oscilloscope, the cell would go bup, 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 bup. And you can see that they tend to segregate in a particular part of the box. We can now take all of that data and take into account how often the animal has been there and things like that. And we produce what the figure you see on the lower right-hand side, which is a color map, which shows you where the cell fired. In blue, you see where the cell doesn't fire, and in red, you see where the cell fires maximally. And in other colors, the cell fires a little bit less. So the cell seems to fire when the animal goes to a particular location, and we call this a place cell. If you look at several of these cells at the same time, what you see is that one fires in the northwest, as this one does, and other fires in the southeast, and so on and so forth. And so if you put all these together, you can imagine that they produce something that you might call a map. They produce a map of space which tells the animal where it is in a particular environment. Okay, well, to get a map, you don't need just a bunch of places. What you really also need to know is the relationship between those places. You need to know where they are in space relative to each other. And so when Nadel and I tried to put this all together into an explanation of what the hippocampus does, we called it the hippocampus as a cognitive map. And we suggested that there must be other kinds of spatial signals in and around the hippocampus. And over the last 40 years now, what's happened in our lab and in many, many other labs in many countries, particularly I have to say in the United States, is the discovery of these other types of cells. Let me give you an idea of what they are. So if you see on the lower right-hand side of the slide here, the lower left-hand side of the slide, you'll see that there are the cells which we call the place cells, and the arrow shows that they are found mostly in the hippocampus itself. If you now move around clockwise and go to the top left part of the screen, you see another kind of spatial cell, which was discovered by Jim Ronk in New York, and which he studied with Bob Muller and John Kuby and Jeff Taube and their colleagues. And this is a head direction cell. This is not a place cell. This is the opposite of a place cell. This is a cell which doesn't care where the animal is. As you can see in the false color map, you get red and yellow all over the place. But if you look to the right, you see in what's called a polar plot, this cell actually prefers a direction in the environment. This cell tells the animal where it's pointing in the environment. So this little blob that you see heading towards the northwest is actually saying that's when the cell fires. It doesn't matter where he is in the environment, it fires there. If you now move again clockwise to the upper right-hand side, then you get these wonderful grid cells, which were discovered by Edvard and May-Britt Moser in Trondheim, which pattern out on the environment, on all of the environments that the animal visits, this grid-like structure. And as you can see, it's a grid-like structure which is symmetrical, and where the distance between the little peaks of firing, so you get not one peak of firing as you get with the place cells, but a whole bunch of peaks, and the distance between them is always constant. It looks as though this is some sort of a metric system. Maybe it's telling the animal how far it's gone in a particular direction, or maybe it's telling the animal what the size of the environment or something like that is. And finally, on the lower right-hand side, you see boundary cells. One of the things we know that the map needs is it needs information about the environment. And it took us a while to realize that one of the types of information that the cells have, and this was discovered by ourselves and also the Mosers at roughly the same time, is there's information about how far the animal is from things in the environment. In this case, it's a wall of the box, but more recently, the Mosers have shown that there are also cells which tell the animal how far it is from objects in the environment. So we have all the information we need to create a map. We have places, and which fly whenever the animal goes to lots of different places in the environment. We have directions from one place to another, grid cells which may be telling you how far apart they are, and information about where the boundaries are in other objects in the environment.
And we all have these place cells in our brains. And so this gets into the sense of space and direction. And I want to talk about that. And I know you have some slides to explain that, but very quickly, because we all live this experience, let's go to our next polling question for the audience. I'm fascinated in the responses here to this question, and then to where you'll take us when you connect it to our hippocampus and what everybody experiences. So our next polling question, if we could pull that up, is a sort of personal one for everybody. When it comes to my sense of direction, which is coming from your hippocampus, at least in part, I have a great sense of direction and navigation. Sometimes I lose my bearings. I need to ask for directions occasionally. I need GPS wherever I go. And you know, John, I think you and I talked about this. My father-in-law, who was a Navy pilot, had the extraordinary ability to go, he could go one place once and always get back to that place, always navigate that. I know other people who could go 100 times and they got to go back to their GPS. What, are we born with some with more place cells than others? Why does that happen? Well, maybe
so you want me to answer that before we do the poll?
We should get people's response first.
Get the poll first, yeah.
All right, let's see what we've got here. But it really is an interesting thing. We've got most of our responses. Let's put the responses up if we can. This is interesting. About 40%, John, say they have a great sense of direction and navigation. 29%, sometimes I lose my bearings. 19%, I ask for directions occasionally. And 11%, I need GPS wherever I go. That's so interesting.
Very good. Yes, and I think that's probably quite representative of the general population. I think the ability and the sense of direction, which we assume comes from the head direction cells, varies across the population. I should say that one of the things that Immanuel Kant suggested was that we did have some understanding of space and construction, the ability to construct space, a priori, before we had any experience. And it's the actual, as we and the Mosers showed some time ago now, it's actually the sense of direction, which is almost what you would call innate. So if we look in animals at the cells and the areas where we know they're going to be these head direction cells, we find that they're there right as early as we can record from them, right before the animal actually has left the nest and has had any experience with the world.
So it's not necessarily, that capacity.
So we don't know that it's genetic or epigenetic,
Right.
or how that comes about, but it certainly is something that's there very, very early on. And we don't know whether people who are good at directions have different genes than others.
I know you have some more slides that take us into this notion of direction and place and how that's happening in our brain. So take us through that, John.
Okay, so let me just tell you about this notion of a cognitive map and flesh it out by using a small section of the center of London and a map of that section. In fact, it's a map of the area we call Bloomsbury, and it's taken from 1830. And what's remarkable about London is many of the things stay exactly the same. My wife and I came to London in the 1960s. We were astonished at how old everything was. Coming from New York, and Montreal, it was astonishing. But it is you can see many of the same streets and many of the same buildings and so on and so forth. So let me tell you how we think the map works by taking an imaginary walk, taking an imaginary tourist on a walk through this section of London, and highlighting some of the artistic and creative people who have lived there and what they've done there. So let's start at University College London, and with Jeremy Bentham here, who was one of the founders of University College and strong supporter of women's rights and the father of utilitarianism. Stroll down from there, directly south along, can you see my cursor? Directly down Gower Street to a house just south of what was then University College London, where Charles Darwin lived from after he came back from the Beagle, and where he wrote the first drafts of The Origin of Species. If we then continue on going south, we go to a small row of houses where Bob Marley lived for a couple of years. He lived mostly in Chelsea, but he did live there in a couple of years. And if we continue, of course, then going straight to the east, we run into a very small house here on the edge of Russell Square, which is where T.S. Eliot worked for most of his working life. He worked at the publisher's Faber, and that's where he wrote most of his important work, The Wasteland, the Four Quartets. We carry on then down to the British Museum, which has been renovated, but still has many of the same features. We realize that that's where Karl Marx spent many, many years writing Das Kapital, which has, of course, had revolutionary effects on many of the countries in the world and caused lots of revolutions. If we carry on now going back north again and go to the southwest corner of Russell Square, then we come across the place where Leo Szilard was crossing around 1933 from the square onto his hotel right across the street, and where he realized that the recent work by Cockcroft and Walcott, which showed where they had split the atom, might be the basis for a chain reaction. He reckoned that if neutrons would create more neutrons when the atoms were split, that would create a chain reaction. And actually, and patented the idea. He gave it to the British government. And he then, of course, when this eventually became clear that this could lead to an atomic bomb, was one of the people who wrote to Roosevelt and prompted him to start the Manhattan Project. If we now continue going north, we come to a series of squares here, and which were the center for the Bloomsbury Group, which included Virginia Woolf and Maynard Keynes, the great economist, who was credited with saving capitalism during the Great Depression. And Virginia Woolf and the Maynard Keynes were a couple of the Bloomsbury Group, who were intellectual powerhouses. There were a whole group of them who came down from Cambridge to Bloomsbury and who generated enormous ideas that have had a great influence on the 20th century. And Dorothy Parker once famously said, the American poet, and Whit once famously said, the Bloomsbury Group lived in squares. They painted in circles because some of them were artists, and they loved in triangles. So we've taken a walk around Bloomsbury, intellectual, creative, artistic Bloomsbury. And what the hippocampus will have done in our tourist will have stored information about the various people who lived in the various places, the places that they visited, how she went from one place to another, and what she observed when she got there.
Well, let me just interrupt if I may. So the hippocampus, when you're doing this cognitive map of Bloomsbury, is doing what exactly? It's
actually storing in the place cells. It's storing the places like University College and the British Museum and the various squares. but it's also storing information about what had happened there, and particularly what you have personally experienced when you went to those places. And it's also storing the spatial relationships between those places.
So this is both our hard drive and our GPS.
Yes, the brain doesn't sort of split things out as far as we can see into processors and memory banks. It actually does everything and stores everything where it does the processing. So it's doing all of that. And it's also doing more than that. And I'll show you on the next slide, because what we want the cognitive map to do is not only tell us where we've been and what's happened there, but we wanted to, trying to move this along. We wanted to tell us how to get around. We wanted to tell us how to navigate. And I have to say, until very recently, it's been very difficult to come up with computational models as to how the hippocampus does that. We have the place cells and the head direction cells and the grid cells and all of these cells, but to put them together into something which would allow you to navigate has been very difficult. And what I'm going to tell you in the next two slides is work that's been done by Jake Ormond in our laboratory, in which he's been able to get animals to navigate to specific locations in an environment, but at the same time be able to ask the place cells at every step of the navigation, where do you think, what's a good direction? As I told you, when we look at place cells and the animals foraging in an open arena, they don't seem to care about directions. It's the head direction cells which care about it. When we start telling them, look, there's a place, oh, there's a goal, all of a sudden they become very interested in directions. And what we see is what's shown on the left of this figure, where the goal is that little octagon, hexagon, and the arrows tell you the direction that the hippocampus as a whole is pointing to as an animal moves around in the environment. So what you can see is, if you look at the
what do you mean? It's
so each cell in the hippocampus has a place it likes to point to, but they're not necessarily the goal. It's only if you look at all the cells together, which we can now do with the new techniques, that you see that the hippocampus as a whole is pointing the animal and the rest of the brain towards the goal. And you can see that by the arrows. So if you look at the bottom, the arrows are tending to point northwards. That means when the animal turns around and surveys the different directions, when he's in the bottom of the maze there, the cell says, oh, I'd like to fire most when you're pointing north. But then if he's in the north, and again, surveying the different directions, now the cell is pointing either towards the east or towards the south. So that the hippocampus is providing information about the direction to go to if you want to get to a goal. And I've shown this as a schematic on the right-hand side using the same map, where let's assume that our tourist wants to go to University College London. And as she moves around to different locations from the southwest to the southeast and then to the northeast, her hippocampus, we assume, is pointing her in the direction of University College. So the arrows change their direction to keep pointing as she moves around in space. Now, so what we think is happening is the hippocampus creates this large red arrow here, which points in the direction of the goal, but it also points in other directions, which are feasible ways to get to the goal. Of course, in the real world, quite often the way that you would like to go is blocked off, as shown by this purple block here. And then, of course, what we would like the hippocampus to do is tell us what's a better of all the other possible directions. And that's in fact what we see as well. What we see is that the hippocampus not only points in the direction of the goal, but it offers advice on how good other directions are. So, for example, if the direction to the goal is not available, then it will say that the direction going straight north shown as shown by this arrow is better than going straight west, as shown by this arrow. As you can see that some of the arrows are bigger than the others, and the bigger the arrow, the better that the direction is to getting to the goal.
So it is our hippocampus that informs us on alternate routes.
That's an
Something
alternate
is
routes. And it
blocked.
says, yeah, you can't go directly there. Then it tells you which are the better of all the possible routes to get there. And of course, we know from, say, from cab drivers, if they know that one route is blocked, they're very good at picking up alternate routes to find their way there. And as
and anybody who's been to London, who's grabbed a London cab, knows
Yep.
that
Yep.
those cab drivers are amazing. Even before GPS, they had something called the knowledge. They had to
Absolutely.
memorize the maze of streets in London. That
Yep.
memorization, that process is taking place in the hippocampus, and this is the firing of place cells.
Yes.
So we train those.
Yes, you train those. And for example, we know from work by Eleanor Maguire that London taxi cab drivers have bigger hippocampuses than, say, London bus drivers or your pedestrians, and that their hippocampuses get bigger the longer they've been driving cabs. The more they need to use this information, the larger the hippocampus gets. And when they stop being cab drivers, it shrinks back to the normal size.
So, I'm interested in, we have Google that remembers our lives, and we have GPS that tells us where we go, and now we don't memorize phone numbers and we don't look at maps anymore. Does reliance on GPS shrink our hippocampus?
Yes, so that's the question that when I went to Google, we had a long discussion as to whether Google was responsible for shrinking our hippocampus. And it raises something which I'm not sure we know the answer to, which is if you're not using a part of the brain for the original function that it was intended, can you use it for something else? A hint about the fact that the answer is probably yes is that it's only the right-sided for most of your life that your right hippocampus is telling you how to get around the world. Unlike a rat, which uses both its hippocampus, humans tend to use their right-hand hippocampus to get around the world. They use the left for storing language. And so we have to imagine that what's happening in the left hippocampus is it's being used to generate narratives of the sort that I just gave you about routes through parts of Bloomsbury, what you did in the past, and so on and so forth. We don't really know very much about that. I myself have speculated a bit about it, but it's an area where we hope to make some more progress in the future. So it could be that, yeah, as you use your GPS more and more, you'll be better at speaking on these conversations.
Yeah, so I know that a lot of the research that you and others have done connecting the hippocampus to our brain health, to dementia, to Alzheimer's, that kind of thing. And let's take a couple minutes talking about that and see if we can get through that quickly and get to audience questions, because there are a lot of them about this and other things. It takes us to our next poll that I'd like to ask the audience to weigh in on. This is a question we probably all think about, and for some it's quite personal, and it's about your own brain health. So if we could put the poll up. Do you have concerns about brain health or dementia? You're not at all concerned. You think about it sometimes, but you figure you'll be okay. You worry a little, you worry a lot. Fascinating question, as I say, and a lot of it probably tied up in genetics as people answer this. John, I know you've connected some of the dots here. Dementia doesn't start or include, just the hippocampus, right? I mean, there's a lot more involved here.
Yep, there certainly is. Do you want to report the poll, or do you
I want to give people a chance to come in. We still got 360 responses so far, so we're getting quite a bit.
Well, let me take the opportunity to say a little bit about this. About 20 years ago, when the Brock's and some of the data shown on the slide began to show the anatomical spread of the toxic molecules that were involved in Alzheimer's, both the amyloid plaques and the tau tangles, and what they showed was that, and I need to close this here because what they showed is shown on the right hand side here on the middle of the screen, is that if you look at these brain pictures here and look at where the red and the orange colors are, they show that at the very earliest stages, when people are just beginning to present with memory problems and finding a little difficulty finding their way around, they that the damage and these toxic molecules are concentrated in the entorhinal cortex and the hippocampus. And that is then, as the disease progresses, the tau particularly spreads from the hippocampus out to the nearby cortical areas, and then eventually, as you see on the right-hand side, to the whole of the cortical mantle. And as it spreads, the areas that are affected correlate with the clinical syndrome. So as it spreads out into areas which are involved, for example, in language and in motor praxic behaviors and things like that, you begin to get deficits, and they show up in the clinical sign.
John, let me just pull up, sorry to interrupt, but let me pull up, because I think this relates directly to what our audience is thinking. Pull up the polling results. And what we see here is 10% are not at all concerned. 33% say, I think about it sometimes, but I think I'm going to be okay. 40% say I worry a little. 17% say I worry a lot. That means 57%, well over half, worry about this. And you got, a third of folks who are thinking about it, but thinking they're okay. So we think about this a lot. I think the bottom line here that I would take away is the vast majority of people are aware of and to some degree concerned about their own brain health. So let me let you get back to.
And they should be, because if you look at the plot on the left-hand side, it's a little bit out of date now, but we knew already in the early part of the 21st century was that if you look at the incidence of Alzheimer's dementia in the population, and what you see is a very rapid increase in dementia with age. It's an age-related disease, and so if you're under 70 years old, you have almost no chances. There are a small number of families who do have a genetic form of it, but as you get older, and particularly when you get to be above 80 and particularly above 90, the incidence is in most countries is at 25% or greater of the population has Alzheimer's. So it's and as the population actually ages, we will find more and more people with dementia. So what we and our colleagues have done and is to try to look at many of these spatial cells in the hippocampus in animal models of Alzheimer's. So we take these, we know what mutant proteins are, and we try to re-express those pathologies in mouse models, and we're getting better. And when I say we, the molecular biologists are getting better all the time. And we look at the place cells, and we see that in some of the animals, not all of them, as the animal ages, these cells begin to be less good at identifying locations. And several of my colleagues are also interested in coming up with tests for the earliest detection of Alzheimer's. If we need to know when people are going to get it before they get it, so that if we're going to have any therapies. And so they're using things like virtual reality and seeing if they can detect the earliest signs of Alzheimer's by people failing to be able to do spatial cognitive tasks. And that's what the slide on the right-hand side is showing.
John, we have so much more that I want to get to, but we have a ton of questions from the audience, and maybe we could go into what I fondly call, from my roots in television, a lightning round here, which is unfair to the questions, because I don't want to boil them down to just a few words. But I'd love to throw some of the questions from the audience at you here and just let you randomly go at that, and then we'll come back. I want to certainly get to, before we conclude here, where you see the future of the field going, because you know it so well. But if I may, Jerome asks, and let's try to just do this as quickly as we can. How does consciousness affect the brain, and how does brain affect consciousness? Big question there.
Yeah, that's a very good question. And I was going to say, despite the fact that when I started many years ago, I was very hopeful with philosophers that we would begin to eventually understand consciousness. I don't really think we've made many inroads into consciousness. Consciousness is clearly a very important property of the brain. It's a function of the brain. it's carrying out an important function, but we're not quite sure exactly what that is. So when you're conscious of that, is very activity in brain areas, sometimes in the higher areas of the cortex, sometimes in the hippocampus. And when you experience acting in the world, then that actually is reflecting the underlying brain activity. But I must say that when you ask me what the future is, hopefully that will be one of the things we'll be able to get a better purchase on in the future.
That's great. Here's a really interesting question. Is there a way to work out this area of the brain? Meaning, as we get older, if we feel our spatial memory or sense of place is less acute, is there anything we can do? It's like going to the gym for your brain.
So and that's an important question. It's true that with normal aging, there is and many studies have shown this, there's a decline in spatial abilities, spatial navigation, spatial memory. The good news is that probably is operating at the functional level. If we look at the number of cells, it used to be thought you were losing cells in your brain all the time as you grew old. It turns out that's not true. And with new
I'm very glad to hear that.
Yes, with new anatomical techniques in general, the number of cells stays roughly the same. There are slight regional differences between those areas. What's happening, though, is that in some areas, particularly in the hippocampus, one part of the hippocampus, the dentate, the cells start acting, they start firing less. They're less active, whereas at other parts, they're firing more. So there are functional changes, and yes, I think I think it's the brain is like a muscle. If you use it, you probably can slow down changes in it.
So if you go traveling and wandering and you use maps and is that if you challenge?
Yeah, but I think there are other things that you can do. We know that brain health depends to some extent on your diet, things like that. Yes, you have to keep working out. And so just being active, but I would guess the evidence isn't very good on this, that, spending your weekend, finding your way around new areas, taking walks in the country is probably a good way to stave off this dysfunction. We don't know exactly how the dysfunction maps out in humans. We've done a lot of studies in animals, particularly Carol Barnes in Arizona has worked a lot on exactly why the cells are changing.
John asks a great question. Where does the brain get its place direction data, e.g. optical nerve? How would a blind animal, John asks, be affected?
Yeah, that's a very good question. And I didn't go into that, obviously, but I did allude to it. So as you're walking around Bloomsbury, you're looking at the buildings, you're looking at the trees, and you're sensing the shape of blocks and things like that. So the hippocampus gets information from almost all the sensory areas of the neocortex. So the main, in animals at least, the main sensory input is vision, probably because you can tell better where you are by looking at objects which are far apart, far away, than things that are close up. But animals which have lost their sight early on in life still have place cells, and they still know how to find their way around. They still can run mazes and things like that. So the hippocampus is smart enough to be able to substitute any modalities for any others and use any information that is given to. It also uses your sense of balance, whether you're turning left and right and things like that. It calls on all of the functions of the rest of the brain.
We have time for just a couple more very quickly. Wendy asks, different cultures, this is really interesting, different cultures organize their cities differently. Does this imply different grid cell structure or organization?
Yeah, so we don't know, of course. I mean, we only have indirect access to the grid structures using functional and magnetic imaging. So I don't know any studies which have looked at different cultures. I give talks in the architecture school at UCL, and I talk about different architects as being kinder to the grid system and others not. So Frank Larry is one of the people I think is not very good for your grid system, and with all of his angles and
it's confusing.
corners. So I don't know about cultures, but I think there is a whole field there which we will be exploring in the future.
I want to, before we hand it back over to Eric in just a moment or two, ask you where this field is going. I know you mentioned that colleagues of yours have been using virtual reality and other fascinating technologies to look deeper into our brain, see grid cells and other things. Where do you see this going, John, over the next 5, 10, maybe 20 years?
So we're benefiting, I have to say, from an influx of people from many other disciplines: physics, computer sciences, molecular biology, many other areas. And so, for example, one of the things we really need to do is not look at single cells, which I spent most of my life doing, but looking at whole groups of cells. So, for example, that a pattern of activity that I showed you, where the neurons were all heading towards a particular goal, that's based on being able to record from quite a few cells in the hippocampus at the same time. And it turns out that the actual signal and the information is at the level of groups of cells and not at single cells. So we need to be able to look at many cells at the same time. For example, if you change where the goal is, then it's new cells which come and point to the direction of the new goal, and you wouldn't be able to see that. So there's a lot of technology. We're now getting tools for actually marking individual cells. We're getting tools, a particular tool called optogenetics, where we can actually identify and activate specific cells independently of their other things. So we're beginning to look at the causal relationships between cells. So I think it's the future is very bright for neuroscience. I think we're going to be able to tell a lot more about the way the brain works at the network level, and not just at the level of individual cells, but we'll be able to look at how the cells interact and how they construct these representations of the outside world and your feelings and hopefully eventually conscious processes.
There's so many great questions we got from the audience. I'm so sorry we didn't have more time to get to more of them, but I'll close with one that I think could prompt a one-word answer, John.
Okay.
Does the Earth's magnetic field play a role in our sense of direction?
So the evidence is for human beings is not very good. People have put helmets on people's head, but the evidence for birds is actually very good. And there's still a question as to exactly what the sensor is. There are some types of magnetic materials in the midbrain. Some people think it's the eye, that when you shine lights in the eye, the rods actually can become magnetically sensitive. So the answer is for many animals, yes. But that feeds into animals in the wild. For humans, the case is still out.
John, thank you so much for all that you have done and all you have helped us understand about this incredible thing called our brain. I mentioned to you earlier, and Eric's back with us, I dedicate this conversation to my 4-month-old grandson, Charlie, because in watching Charlie at 4 months old with his eyes wide and looking and taking in this world, I see the sponge of his brain just absorbing stuff. And
Yes.
as you and I have gotten to know one another and spoken a bit, I think of what's going on in there in his hippocampus and throughout, just pulling in information. It is a marvelous, miraculous thing. And I just want to thank you again for the lifetime that you have brought to helping us understand this thing that makes sense of the world for us and makes us so unique. Thank you, John.
Thank you very much. I feel very privileged to have been able to study it, to tell you. Yeah.
Fabulous. Eric, over to you.
Yeah, so let me thank you, Frank, for a great interview as always, and for this amazing talk, John. And I just want to say that you took a very complicated topic, the most complicated organ in the body, and some amazing research and made it accessible for all of us. So it's really much appreciated. I also like to say that you are an exemplar for why a liberal arts education is so important and why interdisciplinary science is everything today. I mean, you've just shown us that, and I'm not just talking about science and physics and chemistry and biology, I'm talking about philosophy and biology. And I think it's remarkable how much insight you've gained from having that very, very diverse base for what you've been doing. So thank you very much. And I hope everybody on this call realizes how important that liberal arts or liberal education is. I'd like to conclude by thanking everybody, our audience, for phenomenal questions. As Frank's already said, really good questions, digging into this really interesting and long topic. And I want to encourage you to join us again on October 28th. One of our scientists, Alicia Weinberger, will be talking about a brand new instrument she's been building. She's in the middle of building this instrument that's going to be able to look in much more detail at some of the nearby exoplanets, start understanding their atmospheres, whether there's life there, really interesting instruments. So you can't look at these things unless you have the right tools. She's developing the right tools. We heard John talk about tools he developed. These are a different set of tools instead of looking inside to look outside. And so thank you all again for joining us. Thank you, Frank, and thank you very much, John, for a wonderful talk and of course the great work you're doing.
Thank you very much, Eric.
Thank you.